Good morning. I would like to thank all the committee people for organizing this wonderful breast cancer webinar and inviting me for this webinar series. Title of my presentation is Effects of HSP90 inhibitors on triple negative breast cancer. Notch 1 as a target for triple negative breast cancer. I declared that I don't have any conflicts of interest. Triple negative breast cancer accounts for about 10 to 15% of all breast cancers. This cancer is negative for estrogen receptor, progesterone receptor, and HER2 receptors. Worst part of this cancer is it has very limited treatment option and very poor prognosis after standard chemotherapy regimen. We decided to focus on HSP90 protein as a potential target for therapy. It is the most abundant protein expressed in the cells, about 1 to 2% of total cellular protein content. HSP90 is a molecular chaperone that helps guide protein folding. More importantly, overexpression of the protein correlates with poor prognosis in breast cancer and decreased survival rate. Interestingly, TNBCs have been sensitive to HSP90 inhibitor. There are two natural compounds that have been identified as targeting HSP90, called Celastrol and Triptolide. Both these compounds comes from Chinese plant called thunder god vine. This is used in traditional Chinese medicine for more than 100 years. So, first question we asked is, do Celastrol and Triptolide mediate anti-TNBC activity? To address this question, we used 4 TNBC cell lines and primary patient-derived TNBC cells. First, we performed proliferation assay and cell death assay. Next, to determine the stemness, we performed mammosphere assay. And then, for in vivo, we used tumor xenograft experiments. As a first step, we tested for cell proliferation after compound treatment. For this assay, we performed time and dose-dependent treatment of Celastrol and Triptolide. Interestingly, non-cancerous human mammary epithelial cells, HMLE, shows no effect after Celastrol and Triptolide treatment. But TNBC cells significantly decrease in cell proliferation after compound treatment. By using this proliferation data, we have calculated the IC50 value of these compounds. All TNBC cell lines shows 0.5 to 0.6 micromolar of Celastrol and 5 nanomolar of Triptolide. And patient-derived TNBC shows double the concentration. For further experiment, we have used IC50 concentration of these compounds. Next, we performed clonogenicity assay using the IC50 values. We determined the long-term effect of the short-term treatment with compounds. We treated the cells for 24 hours, then changed to regular culture media and allowed the colony to develop. Here is the data. Both Celastrol and Triptolide suppresses colony formation compared to control. Then next, we ask what is the mechanism by which the compound inhibits cell growth. We ask whether there is apoptosis. To address this question, we have used the flow cytometry, imaging, and Western blot technique. Compared to controls, there is a much higher level of apoptotic cell death following Celastrol and Triptolide treatment. For further confirmation, our imaging data shows apoptotic bodies in both Celastrol and Triptolide treatment. In the bottom panel, Western blot data shows that increase in cleaved caspase 3 and pro-apoptotic protein bags And also it shows decrease in anti-apoptotic protein BCL-XL and BCL2 in both the cell lines after celastrol and triptolide treatment. So summary of this data suggests that both celastrol and triptolide inhibit proliferation of TNBC cell lines while inducing apoptotic cell death. Next, we ask whether celastrol and triptolide affect stemness. For this, we performed mammosphere assays and Western blots for stem cell marker proteins. Here is the representative figures for mammosphere growth. There was a dose-dependent reduction in mammosphere growth following treatment with celastrol and triptolide. We also calculated the mammosphere number. We found nice dose-dependent reduction in mammosphere formation in celastrol as well as triptolide. For further confirmation, by using the Western blot analysis, we determined the expression of 3 different stem cell marker proteins, DCLK1, ALDH1, and CD133. In cells grown in 2D culture, there was a reduction of all 3 stem cell marker proteins when compared to control. The similar results were obtained with cells grown in mammosphere. In summary of this data, both celastrol and triptolide inhibit TNBC cells, especially stem cells. How does this compound affect stemness? We decide to look a notch signaling pathway. Why notch signaling pathway? Notch signaling has been shown to be critical for self-renewal of cancer stem cells. The aberrant activation of the notch signaling is associated with breast cancer, and notch and its ligands are overexpressed in human breast cancer. Most importantly, silencing the notch signaling causes growth arrest in both breast cancer stem cells and progenitors. The next question, do celastrol and triptolide affect notch signaling pathway? So we did a Western blots for notch and targeted proteins. Here are the results from the 2D and mammosphere culture. There was a decrease in notch ligand JAGD1, notch functional domain NICD1, and notch downstream protein S1 and A1 in the cells treated with the celastrol and triptolide. Next, we ask, is the inhibition of notch signaling pathway is important for celastrol and triptolide effects on stem cells? To address this question, we overexpressed NICD1 and then grew mammosphere in the presence of celastrol and triptolide. Here is the data. Compared to the untransfected control, celastrol and triptolide did not inhibit mammosphere growth in NICD overexpressing cells. All this data suggests that both celastrol and triptolide suppresses stemness through the inhibition of a notch signaling pathway. The next question is, do celastrol and triptolide inhibit tumor growth in vivo? To address this question, we injected BT20 cells into the flanks of the nude mice and waited for palpable tumors to develop. Then we injected the compounds every day for 3 weeks. The tumor volume from the treated animals are much smaller than the control. We also collected these tumors to determine their weight. The tumor weights from celastrol and triptolide treated mice were also significantly lower than that of control. This suggests that both celastrol and triptolide inhibit tumor xenograft growth in vivo. Next, we check the stem cell markers on notch signaling pathway protein in xenograft tissue. So this data shows the reduction in stem cell marker protein DCLK1, ALDH1, and CD133, and also not signaling pathway protein after Celastrol and Triptolide treatment. So from this overall summary of the data shows that Celastrol and Triptolide inhibits proliferation and colony formation. They induce the apoptotic cell death. Both compounds inhibit mammosphere formation and reduces tumor xenograft in vivo. All this occurs in part through the inhibition of the Notch signaling pathway. So from the summary, we conclude that both Celastrol and Triptolide are potent inhibitor of tumor growth, stem cells, and Notch1 signaling pathway. We believe that both Celastrol and Triptolide may be potential therapeutic or a chemopreventive agent for triple-negative breast cancer. With this, I would like to acknowledge my mentor, Dr. Srikant Anand, and team members who helped me in this project. And also my collaborator, Dr. Roy Jensen, Director of University of Kansas Cancer Center for his generous support, Dr. Scott Weir, Associate Director for Translational Research, and Dr. Andrew Godwin is the Director of Biorepository who is providing a TNBC tissue for this project. Thank you all for your attention. Now I can take the questions. Thank you.